This is a production of Cornell University.
Thank you so much for the kind introduction and for, most importantly, for having me here on this beautiful campus. If you ever visit Texas A&M and you would like to see fall colors, you will never see that. Not now, not later. It's going to be all the same gray in winter time. It's so pretty here. I wish tomorrow when I fly back to Texas, I will take a picture from the plane to show my wife because she misses fall colors. I have quite a bit of slides, but the majority of the slides are simple, so I will go through them fast. And they are showing a bunch of different phenotypes, and that's the point of my presentation today, is to convince you that there is life beyond jasmonic acid. Jasmonic acid is just one of the oxylipins produced by the plants, and plants produce hundreds of different other molecular species of oxylipins, and I would like to leave you with a strong conviction that other oxylipins in plants have a potent physiological array of functions in diverse physiological processes, and especially in defense against pathogens and insects, and even interaction with beneficial microorganisms called colonizing roots. So I would like to show this picture simply because I'm really proud that Texas A&M decided to build a new building for plant pathology and microbiology department, and it's going to be right here. So this is my lab. My lab is quite large, simply because we do a lot of breeding in the field. And I will explain you how that works. So these are all graduate students here, and there is another graduate students, and these are all undergraduates. Okay, so I collaborated with a lot of different people in the world. I was funded by the National Science Foundation for maybe 10 years in a row, and that funding came to collaborate with Nancy Keller at the University of Wisconsin-Madison to study function of oxylipins in maize and oxylipins produced by the fungal pathogen Aspergillus lactis. I collaborated a lot with Ivo Feusner at the University of Göttingen. That's until I went there in 2010 for sabbatical leave, and I now am capable of running all the biochemical analysis that Ivo taught me how to do while in his lab, and now we are doing that in my lab. And in fact, I do not collaborate any longer. I collaborate with these people, but I do not need the services any longer to run phytohormone analysis or volatile organic compounds analysis or other volatiles used by the plants. I started collaboration with a group here with Rebecca Nelson and Judy Coltman, and they are testing our mutants for resistance to northern conolith blight, which is a big problem here in the northern United States, but not a problem in Texas at all. Texas has its own problem, and that's Aspergillus flavus and mycotoxin contamination by that fungus. I collaborated with Chuck Kennerly. He works on Trichoderma virens, as you may know, and the role of Trichoderma virens. Trichoderma virens is a root colonizing entomophyte that induces systemic resistance in the above ground organs, and we just got funded by USDA to study signaling events between the two organisms, and collaborate with Seth Murray and Tom Isaac. It is primarily for field-based testing for mycotoxin contamination. And more recently, I collaborated with Patsy and Sandy Pearson, and that's to study effect of Pseudomonas chloraphis, which also is an entomophyte of wheat and corn that induces systemic resistance and promotes growth. And unfortunately, I do not have time to talk about this research today. So I will give you a brief overview of what I do in my lab and what the idea is behind all of these different projects that we have in the lab. We primarily work with two gene families, lipoxygenases and oxophytodeiinoatidatases. And these genes are required for not only jasmonic acid biosynthesis, but production of other oxylipins. Then I will focus primarily on what we have found so far in terms of the role that oxylipins play in Maize interactions with Fusarium verticilioides, it produces fumonisin, and Aspergillus flavus, it produces aflatoxin. And then I will finish up with showing you the data that suggest that the lipoxygenases and oxylipins that are expressed in roots have a strong, either positive or negative role in the interaction with trichoderma virens and conferring induced systemic resistance in the above-ground orbits. That's also a new project. So what are oxylipins? Oxylipins simply stand for oxygenated lipids that are in plants derived from two polyunsaturated fatty acids. Linoleic acid contains 2 double bonds, 18-2. Linolenic acid contains 3 double bonds, which is 18-3. The resulting products of this primary oxygenation of fatty acids is production of a vast array of oxylipins. And I'll show you the scheme of biosynthesis of these oxylipins. They are suspected, in addition to just monocases, they are suspected to possess potent signaling activities, strong antimicrobial, anti-insecticidal activities. However, we know very little what these molecules are doing in the plants. We know what they do in animal systems. And let me show you what they do in animal systems. This is an inspiration for my lab. So I show only here, there is several reviews. You will never find a review that will show you what oxylipins do in mammalian systems, simply because they don't even call them usually oxylipins. They call them by the class of hormones. And so one of these classes of hormones is called prostaglandins. And there are 6 other large groups of hormones, like lipoxins, thromboxanes, and so on, that are produced by mammalian systems. I will only show you an example of prostaglandins because these molecules are diverse again, and they are produced from arachidonic acid that has 20 carbon chain fatty acid that has 4 double bonds. They produce a subset of prostaglandins. All of these letters and numbers indicate a different molecular species of these prostaglandins. And each of these prostaglandins is produced specifically in specific cells and transported by specific cell transporters and perceived in other different cells by a number of different G-protein coupled receptors. So if you think about jasmonic acid has a single receptor in plants. It's called COI1 receptor, right? So prostaglandins, different molecular species of prostaglandins in mammalian systems have different G-protein coupled receptors and different functions that they convey. And if you look at the list of all the different physiological processes that prostaglandins regulate, it's amazing. They seem to regulate every physiological process in mammalian systems. Aspirin is known to all of you because it's the mode of action of aspirin on us is to prevent biosynthesis of these oxylipins by binding to cyclooxygenases and inhibiting activity of lipoxygenases. And the result of that is downregulation of some of these oxylipins. So, and I will show you some of the data showing that when you knock out some lipoxygenase genes in maize, and all of a sudden, instead of increased susceptibility to pathogens, they display strong increased resistance. So you should think of them as, of that effect, as effect of salicylic acid or aspirin binding to and inhibiting this pathway. So sometimes overproduction of these molecules lead to an array of physiological disorders. So this pathway is so well understood in mammalian system that 60% of the drugs currently available on the United States market target these pathways. Of course, not only synthesis, but also transport, but also specific perception. In fact, my proposal was calculated, because it's a data from this science paper in 2001, he calculated what is current number of drugs that regulate this pathway. And he has estimated 85 of FDA-approved drugs target these pathways in mammalian system. So that's how much we know about them in mammals. So what do we know about plants? Plants produce a bunch of these oxylipins, and they are produced primarily by the lipoxygenase pathway. So what lipoxygenases do is oxidize fatty acid at 2 different positions, 9 or 13, And it's very important that you discern these two types of lipoxygenases because only 13 lipoxygenase is capable of producing eventually jasmonic acid. 9 oxylipins cannot take part in jasmonic acid biosynthesis. They produce what we call 9 oxylipins. So that reaction seems to be simple. Only 4 different products are produced here by lipoxygenase. Yet, the problem with the lipoxygenase pathway and the complexity comes when you take into account that these primary products, hydroperoxy fatty acids, are converted by 7 different sub-branches of the LOX pathway to produce tons of different oxylipins, right? And some of this, I talked to Ivo Feusner, who knows the best oxylipin field in the plants. I asked him one question. So Ivo, how many oxylipins plants produce collectively? Because we measured them in many different species. He estimated 600. And now he believes it's probably more to 700 oxylipins. And every year there is about 4 to 6 reports of new oxylipins being investigated in plants. So the best understood oxylipin in plants, of course, is jasmonic acid. It's produced by the alene oxide synthesis branch of the pathway and involves oxophytodienoid reductases, only few of them, not all of them, to make jasmonic acid and eventual jasmonic acid isoleucine. Now we can measure most of these oxylipins. In my lab, we measure about 75 different oxylipin species, and that is because we purchased this liquid chromatography mass spectrometry instrument. And we are not capable of measuring, unfortunately, all 700 of them. I wish we could, but we only measure what we can get standards for. So my long-term goal of my lab is to decipher functions of oxylipins in maize. And whatever those functions are, we are after what they specific oxylipins are doing for plant responses to stresses, including abiotic stresses. So, and the way we do it is to generate transposon insertional mutants in all of the genes of the oxylipin biosynthesis pathway, if those genes are known. And so in maize, there are 13 lipoxygenase genes, there are 5 alene oxide synthase genes, there are 8 oxophytodienoid reductases, and there are 2 hydroperoxide lyases. Of course, in Arabidopsis, these numbers are about half of that for each of these gene families. So the way we generate these mutants is by taking advantage of the so-called trait utility system for corn available at Pioneer, because that's where I worked as a postdoc. And so Pioneer has created, a long time ago, a couple of dozen years ago, created this huge population of about 48,000 plants, that each one of which was self, that was produced by crossing a new active parent with diverse inbred lines. So that resulted in this large mutagenized population, basically. And what they do at Pioneer is to use gene-specific primers and mutator-specific primers and screen pools of these plants. I don't actually know how many plants are in that pool. And they identify plants or pools and individual plants and individual families that contain insertion of the gene in your insertion in your gene of interest. So it takes them about 2 to 3 weeks. Bob Mill is very good at that. And it takes my lab about 7 back crosses and about 10 years and 2 to 3 generations of graduate students to produce near-isogenic lines at backcross 7 stage that are now apple-to-apple comparison. And honestly, because of how long does it take to generate these mutants, we only published maybe 5% of the mutants that we have generated. So it's a great time to be in my lab because the mutants are generated. You just come in my lab for graduate students and do the research. Okay, so maize genome contains 13 lipoxygenases. They can be grouped roughly into two large subgroups. These are 13 lipoxygenases, and those genes are required for jasmonic acid biosynthesis or green leafy volatile biosynthesis. I personally believe that all these 3 genes are producing GLBs, but in different organs. I will talk today only about LOX10, and LOX7, 8, and 9 are likely to be involved in J biosynthesis. Besides 13 LOXes, the maize genome encodes 6 9 lipoxygenases, one of which is very unique to monocots. We call it LOX12, and I will show you data about this gene. This gene somehow regulates biosynthesis of jasmonic acid by this lipoxygenase. There is this huge potential for crosstalk between the 9 LOXs versus 13 LOX pathway. So this is unusual lipoxygenase. It doesn't even use fatty acid as a substrate. It uses hydroperoxide of fatty acid, which is produced by another lipoxygenase. And what it does, it cleaves all fatty acid into unusual fragments, C13 and C5. And of course, C5, many of you may not have heard of pentanol. So this LOX isoform is likely to be involved specifically in biosynthesis of this volatile pentanol. So today we generated mutants for all of them, but one, for whatever reason, we tried many times to identify mutator insertion in different places in Cold Spring Harbor and Pioneer, then Pioneer again, and we all this failed to identify insertion of any mutator in LOX11 gene. So it's only one left for us to go after. I will talk today about the phenotypes and the function of ZM LOX3, LOX5, and LOX10, and LOX12. All right? If you want to know about any other lipoxygenases, I can talk to you personally. Some of them, like LOX2 and 1, are involved in salt stress tolerance and drought tolerance. LOX4 appear to be required for defense against anthracnose, leaf blight, and stalk rot, but I will not mention about that. LOX5, sharing 90% identity to LOX4, has complete opposite function. It functions in resistance against insects. And in fact, when you knock out that gene, these plants are more resistant to polytropin. And LOX10 is involved in green leafy volatile biosynthesis. Okay, so somehow insertion of mutator elements results in unstable RNA that gets degraded. So we rarely see any transcript using northern blot or RNA-seq now if insertions happen to be in exons of the genes. So let me tell you what we have found so far about the function of lipoxygenases in Maize interactions with Fusarium verticilloides and Aspergillus flavus. So Aspergillus flavus produces a potent carcinogen. In fact, aflatoxin is number 1, the most potent carcinogen produced naturally by microbes. Okay, it's the most potent carcinogen. So it causes cancer development in liver. It's a mutagen, and it also causes immunosuppression in animals. So Fusarium verticilloides produces another mycotoxin called fumonisin, and fumonisin induces programmed cell death, inhibits sphingolipid biosynthesis. In fact, that's how programmed cell death is induced by fumonisin, by binding to ceramide synthase and preventing sphingolipid synthesis and accumulation of precursors of sphingolipids. So the idea about lipoxygenases having something to do with the regulation of mycotoxin production came from Nancy Keller's research, where she showed when she disrupts oxylipin biosynthesis genes in Aspergillus, they called psi factor producing oxygenases. These are named by Nancy as PPOs. Lipoxygenases are also produced, and there is one lipoxygenase Aspergillus flavus that produces other subset of oxylipins. And these psi factors, when you knock them out, they were shown to either inhibit or promote sporogenesis, depending on what spores are regulated, sexual or asexual. And they are potent regulators of mycotoxin biosynthesis. So when you shut down biosynthesis, the psi factors might be steric-1-cystin in Aspergillus nigelens or aflatoxin is inhibited. And and they were also shown to be involved in quorum sensing fungi. So if you look at the chemical structure of plant-derived and fungal-derived oxylipins, you can clearly see the resemblance. Okay, and that suggested that there is potentially a lipid-mediated crosstalk, so that the oxylipins produced by the fungal species can be perceived by the plant to modulate lipid metabolism in the plant. And plant-derived oxylipins may affect the physiological processes of the fungus. We have published a review paper in 2011, which is kind of, it's very complex to show the schemes from the review paper because it's really, really heavy on detail. So, let me tell you what we found so far towards testing that hypothesis, that oxylipins produced by the plant may, in fact, have two different effects on mycotoxins, or either induce mycotoxin synthesis by the fungus or suppress mycotoxin synthesis by the fungus. So in this particular case, when we knock out LOX3, what we have seen right away when we screened seed, production of oxylipins in the seed, we found that 9 HOGs produced from linoleic acid and 9 KOGs were reduced, and 9-HOT, it's an oxylipin derivative from linoleic acid, is completely gone in this LOX3/4 mutant. All right, so they are 9-oxylipin deficient. So LOX3 is not involved in jasmonic acid directly, cannot be. It's 9-lipoxygenase. So when we tested how the fungi grow on the seed, Physaria, we found no difference whatsoever. So fungi don't care in terms of vegetative growth, they don't care about the presence of LOX3 gene in the host. But a big surprise came when we measured fumonism B1 synthesis. All of a sudden, the mutant, when fungus is grown on LOX3 mutant, they produce about 200-fold less mycotoxin. What they suggest that LOX3 is required for induction of mycotoxin biosynthesis. We do not know even now how it works, but that is a fact. That's what the LOX3 function is. Also, without LOX3 in the host, Physaria produce a lot less spores. So, for that reason, we place LOX3 at specifically regulating secondary metabolism and mycotoxin synthesis, and positively regulating by the fungus, and sporogenesis. So, and it appears to have no role in vegetative growth of the fungus in the kernels. So when we tested what it is in LOX3 mutants that may be responsible for such effect on fumonism, so we have ran microarray analysis a long time ago with Darren Brown, still published that study, and we have seen that many other lipoxygenases such as LOX6, LOX9, 10, 11, and OPR7, 8, these two are involved in J biosynthesis, are all overexpressed in this LOX3 seed colonized by Physaria. So we have tested independently some of these genes for which we generated mutants, and what we saw that LOX3 also overexpressed LOX10 and LOX5. That suggests that LOX3 somehow, 9-oxylipins produced by LOX3, suppress expression of these other lipoxygenases. And I will tell you about the phenotypes of these mutants in response to Fusarium and Aspergillus flavus. And later on, we will come back to the same gene to show you how overexpression of these two genes in the roots impact interaction with trichoderma as well. Okay, so when we tested LOX5 mutants, remember LOX3 mutants are colonized equally well when grown on either wild type or the mutant. If you use LOX5, it's clear that LOX5 mutant seed is much more susceptible to Fusarium verticilloides. And probably corresponding to the increased biomass here on LOX5 mutants, we see also increased number of conidia produced. But what was shocking to discover that it had no effect whatsoever on fumonism biosynthesis. So that placed LOX5 as regulating vegetative growth, and probably due to increased vegetative growth, increased conidiation. So LOX5 has specific effect, productive LOX5 has specific effect on the vegetative growth of the fungus. LOX10 mutants, again, in contrast to the LOX3 mutants, LOX10 mutants are heavily colonized by the fungus, and in this case, it's fumonism that was increased. That probably corresponds well to increased fungal growth. And conidia was also increased, but not as strongly as fumonism. So suggesting that LOX10 has more specific effect on fumonism synthesis, unlike LOX5, because the phenotypes of LOX5 mutants and 10, in terms of colonization, are same. So that places LOX10 together with LOX5 as controlling vegetative growth, but LOX10 potentially has stronger effect on inducing mycotoxin synthesis. So this different lipoxygenase that we have just finished generating, just published not long ago, a couple of years ago, we have found that LOX12, again, another 9-lipoxygenase, monocyte-specific lipoxygenase, no dicot contain this gene, it has a very specific effect on defense against Fusarium reticularis via jasmonic acid regulation. So let me show what we found. So LOX12 is here. So it's not a switching block, could not be involved in J biosynthesis. So LOX12 mutant, similarly to 5 and 10, is also heavily colonized by Fusarium. And in this case, Fusarium produces a lot more of both spores and phemones in V1. Probably what the function of LOX12-derived oxylipins is to regulate vegetative growth. So how does LOX12 do it? Okay. Early papers, we were not able to measure oxylipins ourselves in our lab, so we had to send the samples to Germany to be analyzed. But now we can do that analysis, so we did a lot greater analysis of oxylipins produced by this mutant. And so, in addition to seed, LOX12 mutants are a lot more susceptible in mesocotyls to Fusarium colonization. And so when we measured oxylipins, we found that several of the 9 oxylipins, but even some serine oxylipins like this serine KG, not statistically significant though, but 9 KG is specifically reduced in the LOX12 mutant, suggesting that this molecule may be the one involved in resistance, in providing resistance against Fusarium reticularis. So when we tested jasmonic acid isoleucine levels, what we have found that in the wild type seed inoculated by Fusarium, jasmonic acid is increased dramatically. However, not in LOX12 mutants. So somehow this 9 LOX and potentially 9 KOG regulates jasmonic acid biosynthesis. So the question was then, so we submitted a paper just like that, suggesting that role, and then the reviewers say, well, since you published just J-deficient mutants, OPR7A double mutants, why not to test the role of jasmonic acid in resistance to Fusarium reticularis, which was a very valid question, except that LOX12 mutants are so susceptible to Fusarium reticularis that every time we plant it in the field, it doesn't germinate well, because it succumbs to Fusarium and potentially other soil-borne pathogens. It's extremely susceptible, so it took us 1 year to fight with this mutant to grow it in such a way that it doesn't have any Fusarium pre-colonized plants. So, but we were able to do it, and we tested J-deficient mutants as well. By the way, they are also, I will show you this data, they are not able to survive under severe conditions due to extreme susceptibility to pitching. So what we tested is this OPR7/8 double mutants. You have to knock out both of these genes in order to get rid of OPR activity that is required for converting CISA-PDA, the natural substrate for J biosynthesis, into jasmonic acid. These other OPRs, there are 6 of them in maize. We do not know what they're doing. We do not know what they produce, because they use CISA-PDA as a substrate and convert it into some yet unidentified, unknown oxylipin, potential oxylipin signals, and alpha-beta and detoxification of alpha-beta unsaturated carbonates. So when we tested those mutants for resistance against Fusarium, it was really shocking. So we infected the plants before we left the work at about 6 or 7. You come in the morning at 8 o'clock and they already macerated. 2 days later, they just completely collapsed. So that suggests that jasmonic acid is clearly required for defense against Fusarium reticularis, no doubt about that. So, suggested that potentially people who work with Fusarium reticularis and Aspergillus flavus still argue. We are not confident what the lifestyles of these 2 pathogens are. So we still argue whether they are necrotrophs, hemibiotrophs, or, well, not definitely biotrophs, but we do not know if they're hemibiotrophs or necrotrophs. So it suggests, because of requirement of GA for defense against this pathogen that Fusarium verticilloides is likely to be either an ectotroph or at least a hemibiotrough. Okay, OPR7-8 double mutants. It took my postdoc to generate these double mutants about 2 years because he would plant a large field of segregating 1 in 16 population, and none of the plants that he genotyped were double mutants. And instead of coming and talking to me, he says I failed. So next year, he plants, instead of 200 individuals, he plants 5,000 of them. He screens them all, and none of them are double mutants. And then I talked to him, just like, you cannot fail 2 years in a row. What happened? He says, well, I don't know what happened, but there is no double mutant. I said, well, you were supposed to think what that might mean. Of course, what that means that these plants are lethal. So they die for some reason. And sure enough, you can only... Yes?
Damping off or dumping off?
Oh, damping off. Sorry, thank you so much. And dumping off seems appropriate too. Thank you. So yes, we did identify that the culprit in this case was Pythium aristosporum. That's what we saw in Texas fields all the time. Or commercial soils also have Pythium. And so it dies really fast. If you germinate the plants in sterile soil and then transplant them in the field like this, and I don't even know why he did that work, because I know they die from Pythium, but he wanted to see how fast. I said, okay, do it, whatever. He was excited about it. And look how fast they succumb to the infection. It takes only about 10 days to kill 100% of these plants. So they're extremely susceptible. So what was really surprising when we started testing for resistance against Aspergillus flavus and aflatoxin, because that's what Texas farmers care about. They don't care about Fusarium verticilloides. Our problem is aflatoxin. And that's when I lost funding. Because LOX3 mutant that was characterized by extremely low level of contamination by fumonisin are extremely susceptible to Aspergillus flavus. We saw it in the kernel assay, we saw it in the field, we saw that they produce always a much greater level of aflatoxin. So fumonisin down, aflatoxin is up. Colonization by Fusarium is down, is no changed. Flavus just love this seed. So we tested then these other LOXs that are all expressed in LOX3 mutants, and sure enough, we saw the exact opposite. So in contrast to LOX3 mutants, LOX5 mutants were contaminated by aflatoxin, about 5-fold less in every genetic background that we have tested, including the check that breeders use for resistance to aflatoxin. So it's really amazing effect. We tested LOX10 and it was the same. 2006 and 2007, we have every year we test them. We still didn't publish it because I don't understand how it works, because I want to get to the mechanism. But we showed that LOX10 mutants are barely colonized by Aspergillus flavus in kernel assay, and then whatever Aspergillus in fact produces very little aflatoxin. So these two genes appear to be helping the pathogen to colonize, helping the pathogen to produce mycotoxin. So in case of LOX5, I still cannot pinpoint what exactly it produces in terms of what oxylipin it produces. However, we do know for sure that LOX10 is the sole lipoxygenase in the maize genome responsible for green leafy volatile biosynthesis. When you wound a leaf of wild type, you see a spike in the emission of these volatiles. Z3 hexanol, Z3 hexanol, hexanyl acetate, all of them go up, but you cannot identify any traces of these volatiles in LOX10 mutants, suggesting LOX10 is the only gene required for GLV. I wonder now whether GLV have anything to do with making plants susceptible to Aspergillus flavus and to contamination with flavus. So let me switch gears and tell you what we have found for some of these mutants in terms of the interaction with beneficial microorganisms. So we have published a paper before this week. I was invited to publish a paper on long-distance signaling route to shoot. I thought, okay, it's some projects that died a long time ago in 2007, and I never figured out why LOX3 mutants are more resistant in to anthracnose leaf blight, southern corn leaf blight, and I will show you this data. So if you plant LOX3 mutants in the field, they have always shown the same phenotype. They're much more resistant to southern corn leaf blight. They are more resistant to anthracnose leaf blight. They are more resistant to anthracnose stalk rot or fusarium stalk rot. And what was really surprising, that when we tested expression of these genes in response to fungal infection of the leaves, in this case it's Kolomiets the same is true for Globus heterostrophics. This gene is not induced. It doesn't matter how long you measure expression of this gene after infection. This gene is not expressed in the place where it has effect, right? The leaves are resistant to this pathogen. So you would suspect that at least it has to be expressed somehow during infection. It's not. It's not a gene that is expressed in leaves, period. Where it's expressed? It's expressed in roots. That pointed to a potential significance of root expression of these genes on what is happening in the above ground organs in terms of resistance, increased resistance. So the hypothesis then was that LOX3 mutants produce some signal in the roots that are transported from the roots into the leaves and stalks and induce increased resistance. So how to test it? Of course, if you work with dipods, you know what to do in this case. You have LOX3 mutants, you have wild type, you would do the grafting experiment. Unfortunately, for those of you who work with monocots, it's not possible. And it's a huge problem. So then we had to come up with a new method how to do this graft-like experiment. So one time my postdoc was cutting these plants because he was done with them, and when he looked at them, oh my God, they produce so much xylem sap that you can collect milliliters of it within a single day. And I thought, wow, that could be a way for me to know if there is any signal, it has to be present in xylem here. So, but how to apply it? So when you make a tiny incisions 1 centimeters away from each other on these seedlings, and apply twice diluted xylem sap, only 10 microliter, 6 hours before infection, and then infect with Kolomiets graminicula. And what we saw was really interesting. So when you collect xylem sap from wild type and apply it to plants, they are normally infected by Kolomiets, so normal lesions. However, when you take the same plant, the same genotype, and after application of xylem sap collected from LOX3 mutants, these plants were much more resistant to Kolomiets infection, supporting the idea that roots of LOX3 produce constitutively some signal, and you can collect it, and we can measure it, and apply it to the wild plants, and you see this effect. So that also suggested that potentially this gene is regulated by Trichoderma virens that colonizes roots. So remember, when you get rid of LOX3, the plants are more resistant in above-ground organs, but LOX3 is not expressed there. So it's expressed in roots. LOX3 is a negative regulator then of ISR. And if so, then Trichoderma may or may not have effect on this gene or the mutant. So we treated Trichoderma with Trichoderma virens roots of wild type or LOX3 mutants. And of course, in wild type, what you see, Trichoderma induces systemic resistance. So the lesions are much smaller. However, in Trichoderma, you compare the wild type to wild type here. Trichoderma induces increased resistance. In LOX3 mutants, they're already resistant. Doesn't matter how much Trichoderma they have on the roots, they already resistant to Trichoderma. So then hypothesis was that in order for Trichoderma to have an ISR-inducing activity, it must suppress LOX3 expression in the roots. And well, it's easy to propose this hypothesis. I remember like now, when my students were doing this experiment, I thought, oh my God, what if it's not going to work? I will never be able to submit this paper. And I was shocked how beautifully it worked. So then, and what we did, we used a delta SM1 mutant of Trichoderma. Without this small peptide, it's a secreted peptide. When you knock out that gene for SM1, these plants are not able to induce, here's knockout mutants. They were not able to induce systemic resistance in the leaves. When you overexpress, the lesions were small. So this SM1 secreted protein is required for inducing systemic resistance in the leaves. So we use this mutant and compare it to the effect of this mutation versus wild type on expression of LOX3 in roots. And what we saw, that wild type indeed, like I really hoped it would happen. When wild type trichoderma infects roots, LOX3 expression is repressed. However, LOX3 cannot be repressed when you use this ISR deficient mutants. So that places LOX3 as one of the targets for SM1. One function of SM1, fungal gene and fungal protein, is to suppress expression of LOX3. Then the question was, what it is in LOX3 that makes it so crazy that it produces this ISR signal all the time? So we have tested expression of all the lipoxygenases for which we have mutants or some other genes, and what we saw is incredible. So if you look at 9 lipoxygenase expression in the roots compared to wild type, there is 20-fold induction overexpression of LOX4 in the LOX3 mutants. LOX5 was overexpressed, LOX12 was overexpressed, and a bunch of other genes. And look at this ACC-CHS or OPR8, for example, 22-fold, 24-fold, 20-fold, aldehyde cyclase, 48-fold overexpression in the roots. So the roots of LOX3 overexpress a whole bunch of this other oxylipin biosynthesis and ethylene biosynthesis genes, which suggests that jasmonic acid, 9 oxylipins, and ethylene may be the reason why these mutants are more resistant to foliar pathogens. And that signal is produced in LOX3 mutants constitutively rather than in inducible manner. So we tested LOX10 just because we knew exactly what it is doing in terms of biochemistry. It's required for GLB biosynthesis, but also because we found something really unique about LOX10. We had a project to localize subcellularly where LOX10 is found in the cell. So we produced transgenic plants with the help of Dave Jackson from Cold Spring Harbor, and they produce transgenic plants that express YFP tag LOX10. And they looked everywhere where LOX10 is, and found that LOX10 is found in, it's found in chloroplast, and they also identified some other organelles. They still do not know what it is. But what was really surprising to see is that LOX10 seems to stream in both phloem and xylem because it moves in opposite directions. So LOX10 directly as a protein is secreted into phloem and xylem. So that made LOX10 a perfect candidate to be relevant to long distance signaling, right? So then we tested LOX10 mutants for ISR in response to trichoderma pollinization. So when you have wild type, SMPC in this case, without trichoderma, they are normally susceptible. The addition of trichoderma to the roots makes it more resistant to polytotricum infection. LOX10 mutants are very surprising because GLBs are believed to be required for defense against pathogens. We did not see it. In fact, these mutants were more resistant not only in the leaf. LOX10 mutants were resistant in the stem to anthracnose leaf blight, to Fusarium stalk rot, to Fusarium stalk rot. They were resistant to everything we tested. But immediately after you treat LOX10 mutants with trichoderma, these plants become all of a sudden completely susceptible to polytotricum infection. Not only they have lost induced systemic resistance, they gain some interesting phenotype. I call it induced systemic susceptibility. Because remember, these plants are resistant without trichoderma. However, they are a lot more susceptible when trichoderma is around. So that suggests that LOX10 is relevant to it. And we tested other mutants in that list. And at least we found exactly the same phenotype for LOX12 mutation. So LOX12 mutants are normally, there is no change in resistance levels. But when plant is colonized by trichoderma, they become a lot more susceptible. So we are now, we were funded by USDA to identify these long distance signals And we are utilizing the xylem set that we can collect massively, and we are profiling oxylipins, hormones, and other metabolites. And we already have several candidates that needs to be tested. And those candidates are, by the way, are not jasmonic acid. It's totally against the grain because jasmonic acid is believed to be required for ISR in diploid species, not in maize. When we used OPR7, 8 GA deficient mutants, they are still capable of responding to trichodermal colonization by increased resistance in the stem and in the leaf. So GA is not the long distance signal. It's some other oxylipins produced by 9-lep oxygenases or precursors of GA that are required for induced systemic resistance. So with that, I would like to invite your questions. By the way,
This has been a production of Cornell University, on the web at cornell.edu.